Case Western Reserve University's Institute for the Science of Origins proudly presents the Origin Science Scholars Program. The Institute advances the scientific understanding and application of origins and evolution of human and natural systems. The Origin Science Scholars Lectures are presented with the assistance of Case Western Reserve University's Siegel Lifelong Learning Program, College of Arts and Sciences, and Media Vision. Tonight it's my pleasure to introduce Carlos Crespo Hernandez, who is Associate Professor in the Department of Chemistry at Case Western Reserve University and co-director of the Center for Chemical Dynamics. He earned his bachelor's and PhD from the University of Puerto Rico, and his research group focuses on understanding light-induced phenomena in molecules. And tonight he'll be talking to us about sunlight and the molecular origin of life. And this talk is co-sponsored by the Emeriti Academy.
Good evening, everyone. I first want to thank Glenn and Patricia for inviting me to this series, and thank you everyone that is here for being present today. This presentation is divided in three parts. In the first part, I will review some basic ideas about the molecular origins of light on Earth. I will then summarize several groundbreaking research suggesting why nature may have selected the DNA and RNA bases as the building block of life. I will then discuss how primordial RNA and DNA polymers may have been able to survive the harsh ultraviolet radiation conditions that were present on early Earth. Finally, I will discuss how these primordial polymers may have used ultraviolet radiation to self-repair UV-induced chemical damage in the polymers. Besides human curiosity, the development of scientific ideas about the origin of life may have originated of how life may have originated in our planet is important because it allows us to think more informed ways on what condition life could have exist on other planets. For instance, did the sun play a central role in shaping the molecular composition of the bases on early Earth? And will life in other planets also use these molecules are the building block of the genetic materials? For simplicity, throughout this presentation, I will be using the suffix mer to signify a repeat RNA or DNA unit. I will often represent a RNA or DNA unit using a small brick. For instance, a dimer is composed of two bases, while a polymer is composed of multiple bases or bricks, as you can see in this line. Let's first review some prevalent ideas about the molecular origins of life. Geologists estimate that the Earth was formed about 4.6 billion years ago. The estimate comes from measuring the ages of the oldest rocks on Earth, moon rocks, as well as meteorites using radiometric dating. For many millions of years, our planet was bombarded by asteroids and other celestial objects, and the temperatures were too high to support life on Earth. The earliest evidence of life comes from fossils discovered in the West Australia, dating back about 3.5 billion years ago. These fossils are known as stromatolites and are generally formed by the growth of layer upon layer of single-cell microbes. Therefore, it's currently thought that continual life on Earth began about 4 billion years ago. However, it should be highlighted that the lack of earlier fossil evidence makes identifying the exact times of life origins extremely difficult. Ancestors of cyanobacteria may have flourished on Earth as early as 3 billion years ago. The evolution of photosynthesis allows cyanobacteria to convert some radiation into chemical energy. the formation of oxygen molecules as byproduct of photosynthesis eventually transform the Earth's atmosphere and thus paving the way for the growth of biodiversity on the planet. The available evidence suggests that the ozone layer was first formed approximately 2 billion years ago as a consequence of a so-called the Great Oxidation Event. Of great relevance to this presentation, the dramatic change in the transmission of ultraviolet radiation in the atmosphere happened more than 2 billion years after the first appearance of living organisms and the prebiotic era that came before it. So, how then may life have emerged? In 1920, Oparin and Haldane independently proposed that life emerged gradually from inorganic molecules in which building blocks such as nucleotides and amino acids form first, then later combining to make polymers, complex polymers. The details of the model are controversial, but the basic idea of a stepwise spontaneous formation of simple, then more complex biopolymers, following but self-sustaining biological assembly, is still at the core of much origin of light hypothesis today. In 1953, Stanley Miller and Harold Allray did an experiment to test Oparin and Haldane's idea. The Miller-Allray experiment provides the first evidence that the organic molecules needed for life can be formed from inorganic components. Researchers now think that the atmosphere of the early Earth was different from that simulated by Miller and Ulrich. However, recent studies have shown that RNA nucleotides can be formed as, from inorganic components under conditions thought to resemble those of early Earth. From these experiments, it is reasonable to imagine that at least some of the life building blocks could have been formed abiotically on early Earth. However, exactly how and under what conditions life on Earth originate remains an open question. There are currently two prevalent theories about the origin of life on Earth. Some scientists support the RNA war hypothesis, in which self-replicating RNA polymers are thought to have formed first. According to this theory, RNA, with its ability to store information and perform catalytic reaction, came first, then DNA and proteins. On the other hand, other researchers favor the metabolism first hypothesis, in which metabolic networks came before RNA and DNA. Regardless of which theory of the origin of life we support, an important question that needs to be answered is how could the RNA and DNA bases survive the harsh ultraviolet radiation conditions of early Earth and assemble into polymers.
What caused the ozone layer to form?
It's believed that the cyanobacteria and the beginnings of photosynthesis generate oxygen species, which then, through the interaction with the sun, generate ozone and create the ozone layer. What role does it have on the beginning of life would be part of my second presentation. So if you could wait, I will appreciate it.
Can you just say briefly why it's damaging to not have it for life today?
Yeah, so the ozone layer, as we will see, chills some of the high energy UV radiation from the sun. And it is believed that that helps to biomolecules to prosper and evolve to the beginnings of life on Earth. We will see in the next section that the DNA bases absorb very strongly in the region where the high energy radiation from the sun reached the surface before ozone layer. And so it begs the question, how and why the DNA bases were selected in the first place as the building block of life, and I will try to answer that question.
We seem to have an anachronism of several billion years. Before there was ozone, one would think that there had to be oxygen. The oxygen would have to come from either photosynthesis or something equivalent to it from plant matter in general or bacterial matter in general, which means that there was life before there was an ozone layer. Now, if there was life before the ozone layer, how did the life survive the ultraviolet radiation for 2 billion years until there was an ozone layer?
Yes, that's exactly right, and as I said, that will be part of my next argument. So it's really amazing how the DNA bases which absorb ultraviolet radiation were used to building blocks of life, given the fact that the oxygen and ozone layer were first formed 2 billion years after the formation of cyanobacteria. I'm going to provide some hypotheses of why we think that the DNA bases were selected as the building blocks, and why the sun energy actually will play a very important role in this event.
Thank you for joining us. You've been watching Professor Carlos Crespo Hernandez discussing abiogenesis, the molecular origin of life. For more information on the Origin Science Scholars Program, please visit the Institute's website at origins.case.edu. In the next part of the talk, Professor Crespo will discuss how ultraviolet light from the sun may have played a key role in determining that only 5 bases are incorporated in the RNA and DNA molecules that life on Earth universally depends on. Now, back to the talk.
Okay, so in the second part of the talk, as I said, I'm going to be talking about the sunlight and the selection of the building block of life. Today's biomolecules are thought to originate from simpler inorganic and organic molecules that either arose spontaneously on early Earth or were delivered by comets or asteroids from chemical inventories present elsewhere in the solar system. A period of chemical evolution may have shaped the progression from simple to complex molecules capable of fulfilling biological functions. However, understanding the events that constitute the molecular origin of life is a grand challenge. As shown in this slide, researchers seeking to identify the molecular ancestor of RNA frequently focus on how easy it is to make the bases and their precursor, starting from chemical compounds presumed to have been present on early Earth. Given the large number of organic compounds available during the prebiotic era, it begs the question as to why only four bases each make up RNA and DNA polymers, and whether the sun radiation provided a key role in their selection. This figure shows the radiation spectrum from the sun, both at the top of the Earth's surface and at the sea level. As sunlight passes through the atmosphere, sun radiation is absorbed by small molecules like water, carbon dioxide, and the ozone that have a specific absorption band as shown in the figure. That will be the red spectra there. In particular, ultraviolet radiation from the sun is often invoked as an energy source that could have driven prebiotic chemical synthesis. However, ultraviolet radiation also has enough energy to both make and break chemical bonds. As shown in this figure, all the DNA bases and RNA bases absorb ultraviolet radiation below 300 very efficiently. This ultraviolet radiation is nowadays blocked by the ozone layer, but this was not the case during the prebiotic era. These facts suggest that any prebiotic molecule present in high concentration during the primordial soup may have been molecular survivors of the high degree of resistance to ultraviolet radiation. This idea brings us to the following hypothesis. Chemical stability to ultraviolet radiation may have played an important role in the selection of the building block of life during the prebiotic era. This so-called photostability to ultraviolet radiation is the ability of a molecule that absorbs ultraviolet radiation to avoid chemical change by returning to the initial state it was before absorbing the radiation. From a molecule to a high energy, to be highly photostable, it must absorb energy before the chemical transformation takes place. To the contrary, photoreactivity is defined as the ability of a molecule that absorbs ultraviolet radiation to produce a chemical change. In a photoreactivity event, the final state is different from the initial state, which results in the formation of a new molecule or product. Therefore, photostability to ultraviolet radiation may have shaped organic chemical inventories, and this molecular property can potentially provide new insights into the early events that led to today's biomolecules. The need for photostability will have persisted throughout the whole period of chemical evolution and will have been especially important in the absence of sophisticated repair enzymes. In general, the faster a molecule returns to the initial state, the more photostable it is. To measure the time that it takes for a molecule that absorbs ultraviolet radiation to return to the initial state, researchers use the so-called pump-probe transient absorption technique. In this technique, the molecules are first promoted to the high energy state and by absorption of a laser pulse in the ultraviolet region. Then a second laser pulse is used to detect the time that it takes for the high energy state to return to the original state of the molecule, or the ground state. This figure shows the experimental results for the return of the high energy state in the DNA and RNA bases to the original state after absorption of ultraviolet radiation. As the scheme to the right of this figure depicts, the high energy state shown as a blue brick, while the origin brick represents the conversion of the absorbed ultraviolet radiation into internal energy of the molecule, followed by heat transfer of the excess energy in the molecule to the surrounding sorbent. So first, the molecule absorbs the ultraviolet radiation, forming the S1 state, or what I call the high energy state, from which the energy relaxes in hundreds of femtoseconds. 1 femtosecond is 10 to the minus 15 seconds, forming a high energy molecule in the ground state. That excess energy of the molecule in the ground state is then released as a heat to the environment, therefore returning the molecule to its original state without causing any chemistry or chemical damage. This extremely fast conversion of the absorbed ultraviolet radiation into heat provides all the bases with a high degree of photostability. A closer inspection of the molecular structure of the RNA and DNA, as shown in this scheme, suggests that bases may have originated from one or more common molecular ancestors. In the figure, X and Y denote common functional groups among the bases. A systematic investigation has shown that the position at which the functional groups are located has also an important influence on the photostability or photoreactivity of the purine base. As can be seen in this scheme, the functionalization in the position 6 and 2 of the purine molecule maintain photostability. If the functional group in the position 6 is removed, the molecule becomes highly reactive. So nature would not like to use those as a building blocks. Similarly, the same phenomenon is observed for the modified pyrimidine bases. In the pyrimidine base, as shown to the high left side, the position 4 and the position 2 are equivalent to the position 6 and position 2 in the purine base. And when the functional group is removed or is absent of the position 4, the molecules, the pyrimidine molecules become highly reactive, while when they have functionalization in both the position 4 and 2, they become highly photostable. This investigation suggests that the position at which the chemical groups are located may have played a critical role in the selection of the building block of life from a large chemical inventory of modified bases and other organic compounds during the prebiotic era. In fact, chemical modification of the bases usually results in a significantly more photoreactive molecules. This plot shows the increasing photoreactivity with the different functionalization of the bases. As you can see, the blue dots represent the RNA and the DNA bases, while the red dots represent the modified bases. All the modified bases show high photoreactivity, while the RNA and DNA bases are photostable to ultraviolet radiation. These figures summarize the main results presented in this section. It is argued that the competition between photoassisted formation and photodestruction determines the kind of complex molecules that can be formed. With the passage of time, represented in this picture by the orange arrow, extended periods of ultraviolet processing may have caused the chemical inventory to become enhanced with intrinsically photostable molecules that can be used to form more complex biomolecules.
Obviously, the atmosphere of the Earth has changed dramatically since the early times when what you're describing occurred. Are there any conditions whereby today inorganic material can, without a laboratory doing it, be created into living organisms?
That's an excellent question. So nowadays we have the ozone layer, which blocks most of the high energy radiation that I'm suggesting here in this cartoon, that was used to filter out the many organic molecules available and maintain those that were highly photostable. This happened more than 2 billion years ago, before the creation of the ozone layer, or this is what our hypothesis is. And so currently there is no creation of living matter that I'm aware of with the radiation energy that arrived to the Earth's surface after going through the atmosphere.
Do you postulate that all the raw materials were here when the Earth was formed, that the whole periodic table was just sitting waiting to be used?
No, so there are two possibilities, which most likely both are possible. One is that from the Earth components in organic matter, small molecules, life was created eventually because of the interaction with the sun. The other theory is that many of these organic molecules came in asteroids from the universe and then land in the Earth. That's the cartoon of the rock in the left side of the panel. I'm not arguing how the inorganic matters arrive. We actually cannot prove one way or another, even though many meteorites that have been analyzed show evidence of organic molecules, like including DNA bases and RNA bases. So definitely these building blocks can be formed in the outside the Earth, but they could also have been formed in the Earth, and so what I'm arguing is that the ultraviolet radiation available provide the energy and like a selection mechanism to basically select the RNA and the DNA bases as the building block of life. And the reason for that is, as I said, because they are highly photostable. They can absorb this high energy radiation. We are talking about 4.7 to 5 electron volts of energy, which is enough to break any chemical bond of organic molecule. And they have this ability to dissipate the energy very quickly as a heat to the environment, which is actually the way that sunblocks work. So sunblocks absorb electronic energy or high energy radiation and dissipate them very quickly as a heat to the environment. So the DNA and RNA bases could be considered as a natural sunblocks in the prebiotic era.
So just to clarify, all of the elements were here, but they might not have turned into molecules here.
Yes, the elements of the periodic tables most likely were here and formed billions of years ago. Whether the small molecules that we call inorganic molecules composed of 2 and 3 atoms like water, carbon dioxide, methane, and others, were formed through the Earth's process or came from the outside, both are possible. What we are basically arguing is that these molecules then somehow were selected to form the biomolecules on the origin of life. And we think that the sun, the ultraviolet radiation in particular, play a very important role in their selection because they are stable to this type of radiation.
It seems like we often hear the story that a lot of these processes are by chance. And you're presenting a story where a lot of it is actually selected for long before life originated, rather than there being as big an element of chance as we're often led to believe.
Yes, and I want to stress that this is just an hypothesis as many others, but it's an hypothesis based on many collection of experiments that show that these molecules in particular, the DNA and RNA bases, are highly stable to high energy radiation. And so if we think of a simplistic way of the original life as a soup of organic molecules in the universe or in the Earth, then the ultraviolet radiation assisted in the selecting of the bases that could survive the prebiotic era and being used as a building block of life.
So from this point of view, evolution begins before life. So the set of chemicals that are available actually evolves before metabolic processes and replication evolves.
Yes, but I want to, so I call chemical evolution, and I do that purposely to differentiate between biological evolution, which is the Darwin evolution. So this is a selection process, which is all abiotic, and so there is no life form yet. It's just the organic molecules available that, with the interaction of the UV light, are being selected as the building blocks. But yes, I will call that, and the community call it, the chemical evolution, to differentiate between the Darwin biological evolution that we are now.
Could you go back to the Miller-Urey picture? You know, I get a preview of these talks, right? This is really what he's saying here I have never heard before about the extent to which there was selection going on long before anything you would call life. All right, yeah, so in this picture, which is one that everybody tends to see, in high school, right? people often, if you could just explain what's going on there, and people make sometimes a criticism of this, that, well, we don't know exactly what was in the atmosphere at the time, so maybe this is invalid. But could you explain what's so important about this, even if it wasn't the exact chemicals?
Yeah, so in this experiment, what Miller and Ori performed is that they put a vessel with water, which is shown at the bottom, and they inject a mixture of gases that they thought to be available during the prebiotic era. In order to initiate the chemistry or the reaction, they provide electric shock to simulate lighting, and by using these conditions, they were able to generate in the big balloon to the left molecules such as DNA and RNA and amino acids. And so this experiment shows that if you add a mixture of small inorganic molecules and provide water and provide some kind of energy, you can generate the building blocks like the DNA and RNA bases by this process. The problem with the experiment is that the scientists use a very reductive atmosphere or conditions that nowadays is believed that were not the exact conditions that the Earth was present in early times. However, as I mentioned, there have been large numbers of more recent experiments, as recently as last year, where science have been simulating, all this is in the lab, conditions that are closely related to the conditions that now we know that were available during the prebiotic era, and they can still generate the nucleobases and nucleotides and amino acid form. And so really this experiment just, the importance, besides historical importance, the major importance of this experiment is that it was able to show for the first time that you can take a mixture of small molecules, inorganic molecules, provide energy, and generate the DNA and RNA bases and the amino acids that are the building blocks of the more complex polymers. So that's the key of this experiment and why the community use it so heavily.
I'm still trying to get my head around the photoreactive organic compounds and the photostable compounds. Do you need photoreactive first and then absorbing energy from whatever the source and drive it to photostable? Is that the, or am I not understanding this correctly?
Let me just go back to this cartoon and try to explain this again. So basically, you go from one state that I call the original state, where the molecule is before absorbing the ultraviolet radiation. That will be the S0 in this plot. In a photostable event, in a molecule that is photostable, they absorb ultraviolet radiation, as shown in the left picture in this slide, and they are promoted to what I call a high energy state, or S1 in this cartoon, okay, in this figure. Then molecules that are photostable, they have the ability to take this energy and dissipate very fast, very quickly back to the original state in the form of heat. So the energy has to be transformed, energy has to be conserved, so the energy is transformed from electronic or ultraviolet radiation into heat transfer to the environment. So the photostable molecules, the initial state is the same as the final state when they absorb ultraviolet radiation. In a molecule that is highly reactive, what happens is that the molecule also goes from the original state to a final state, but then in the final state they do chemistry. So instead of releasing this energy as a heat, they use the energy to make a chemical transformation and forming a new molecule, which is so that the initial state of the molecule is different from the final state, meaning that you form, from one molecule, you form another molecule. Okay? And so that's the main difference. And so when you have this pool of molecules in the prebiotic era, those that were photostable, those that can release the excess energy as a heat and maintain themselves, they will go along through the process, while those that are transformed to some new chemicals, then they cannot be used later on in the process. So the ultraviolet radiation provide the energy to make chemical transformation to potentially molecules that are more stable, and then those molecules that are more stable nature can use it for making biopolymers.
Okay, so basically, you want the ones that are photoreactive so that they can create the organic compounds that you need for light? I mean,
Yes,
I understand this, I just don't know what the next step is after that.
That's an excellent question. So nature have a chemical pool, okay? So all these molecules are present. The sun is being used to selectively separate those that are more photostable from those that are highly reactive. And so the molecules that are highly reactive will be transformed into something new. If that new molecule is still reactive, it will continue transforming. But eventually, these take from a big pool of many molecules, of organic molecules, it concentrate those molecules that are photostable. And then we think that those can be used to create the polymers and the building blocks. So, which is the second part of the talk is, if these molecules were stable, it makes sense to use it as a building block of the DNA and RNA polymers. The question then is these polymers are also photostable, because there are different interactions, as we will see, that you have in a single strand and double strand polymer that are not found in the single bases.
So if I understand correctly, the issue here is that the one on the left, it absorbs the ultraviolet, and then it lets it go and it returns to the state, so it can be used over and over and over again that way, and that's what gives stability to the RNA or DNA. And the one on the right, we tend to think of what we want to see as more complexity generated, because that's what we think about with life, when in fact it's the one on the left that doesn't do something that's unusual, whereas on the right-hand side it gets sort of cooked, and then it doesn't go back to what it was doing.
That's basically correct. So again, the molecules that are photostable, they just go to a high energy state, they come back to the same state, so they do not transform chemically. They are very stable, very robust, that can be used as a building blocks. Those that are highly reactive will transform into a new chemical species, or a new chemical molecule, which then the sun radiation can again filter out. If it's highly reactive, it will continue chemically evolving to form something that is stable. And those molecules that are stable is what we see in particular the DNA and RNA bases, which are stable, that play an important role in the formation of the polymer. Another thing that I want to highlight in this process is that this experimental data that have been shown by many scientists now, that molecules that are very similar, so if you look at this plot, the same six-member ring that you see in the top is in the three molecules, but you have these groups X and Y that are different. And it turned out that when you have the functional groups correctly for actually form wax and acrylic base pairing, so when you have a carbonyl in the position 4 and an amino group in the position 2, these molecules actually are highly photostable. And so in the sequence of the event, you can think of having many of these molecules, and then the sun selecting them to form the RNA and DNA bases, which happens to also be self-complementary to each other, forming the base pair and interaction that are needed to make the polymers. And so that's what we be discussing in the last final section.
We hope you've been enjoying the Origin Science Scholars Program with Professor Carlos Crespo Hernandez. Professor Crespo is Associate Professor of Chemistry at Case Western Reserve University and co-director of the Center for Chemical Dynamics. In the second part of our talk, we learned how the ability of certain base pairs to quickly shed the energy they absorb from sunlight may explain why life on Earth makes such consistent use of them. In our final segment, Professor Crespo will talk about how the behavior of those bases is affected by their incorporation into the DNA and RNA molecules. Now, back to our talk.
So, as I mentioned and argue in the previous section, the intrinsic resistance of the RNA and DNA bases to chemical damage by ultraviolet radiation suggests that they could have been accumulated to more significant degrees than less stable molecules on the early Earth. Then the question that now is important to answer is whether this remarkable photostability of the bases extend to single and double-stranded RNA and DNA polymers. In this final of the part of this presentation, I argue that constructing polymers from photostable molecules make use of more abundant building blocks in the primordial soup that are constantly reshaping by ultraviolet radiation. In the plot here, I chose the representation of the two important features of a single strand and a double strand. In particular, single strand are formed by base stacking interactions as shown in the right figure, while double helix are formed by both base pairing and base stacking interactions. Scientists have dissected both kinds of interactions to learn how and whether these interactions, these new interactions that were not found in the building blocks, affect the photostability and the photoreactivity of the single strand and double strand. In this example shown here, science have made the Watson and Crick base pair, which are shown to the right in this figure, and they have studied their photostability or the interaction with the ultraviolet radiation. It has been shown that from many possible hydrogen base pairing interaction that the combination of these bases can form, and they form in nature, only those that are Watson and Crick base pair shows a highly degree of photostability. And that is shown here with the scheme to the left, and the mechanism or the process by which this hydrogen bonding Watson and Crick base pair maintain the photostability is that they absorb ultraviolet radiation. They, in the high energy state, they shuffle or they transfer a hydrogen from one molecule to the other, forming the species in the right, top right of the figure, and then the base pair shuffle the hydrogen atom back and decay to the original state. So it's a hydrogen forward and backward transfer, which is easily available because the bases are base pair by hydrogen bonding, so they just transfer a proton, a hydrogen back and forward, and that way they sustain stability. Instead of making a chemical decomposition, they maintain highly photostable. In terms of the photostability of single-stranded DNA polymers, this plot here at the bottom, it shows the time that it takes for the high energy state that is formed by absorbing ultraviolet radiation to return back to the original state for both a single-stranded adenine polymer and for adenine or AMP itself. You can see that there is a really dramatic difference between both processes, and the question is then how and why, if the high energy state survive for 3 orders of magnitude, whether this is a photostable molecule. So from many experimental evidence that have been accumulated, this mechanism or scheme shown to the left in this figure explain how and why the single-stranded polymer in where the bases are stuck to each other can survive the ultraviolet radiation. So they absorb ultraviolet radiation, and when two bases that are stuck to each other, two neighbor bases, an electron is transferred from one of the base to another, shown in the top right part of the figure, forming what is called a charge-separate state, represented by the green and yellow blocks in the figure. This charge-separate state then can recombine back, so the electron can come back to the original state, forming again the original molecule and a very photostable molecule. Okay, so it's different from what we thought for the photostability of a single base, for the photostability of a base pair, which involves hydrogen bonding. And here, when you have stack, it formed by electron transfer. But the final outcome is that they have find a way to survive this high energy radiation and maintain a photostability in the polymer. So let's now look at the single strand of timing, and these are just examples. There is a lot of evidence for different combination of bases. I'm showing two classic examples where the single strand of adenine will then form a double strand with the complementary single strand of thymine. And so here, maybe somewhat surprisingly, what we see is that both in the single strand thymidine polymer and in the base, both return to the original state in very fast time scales, and actually in hundreds of femtoseconds. And the question is why, no? And so what scientists have shown is that the single-stranded polymer of thymidine bases, the bases are not stacked. They are just randomly distributed, which is shown in this cartoon by the bases just looking at different directions. And so when this single-stranded polymer of thymidine bases absorb ultraviolet radiation, they go to a high energy state, but they behave as the monomer, as the same as the single thymine bases, because there are not stacking interaction, and they just dissipate the energy as a heat, then maintaining the high photostability in the single strand. So the question then is, what happens if now we put both single strands together and form the double helix? In this case, composed of either, as shown in the figure here in the bottom, as either as a full single strand of adenine bases or a full single strand of thymidine bases. That's shown in the color purple. There is also a second double strand that is shown here, in which now you have in a same single strand an adenine followed by a thymidine repetitively, and the other strand also an adenine followed by a thymidine. Both of these double strands can both of these polymers can self-complement, forming the double helix. And so what is shown in this figure is that the time scale that it takes for the high energy state to go back and return to the original state significantly varies by where two bases are being stacked to each other. However, in both cases, the state return to the original state. And so this is shown with this cartoon in the left. So in the double helix, you have the absorption of ultraviolet radiation, and now what is happening is that both an electron and a hydrogen are simultaneously transferred to the another base, as shown by the blue, red, and yellow bricks in the figure, forming again a charge-separate state as in the single-strand polymer, which then recombine in hundreds of picoseconds back to the original state, and that way it maintains the photostability. So basically in the double strand, it's a combination of the mechanism that we thought for the hydrogen bonding bases and the stacking interaction. So they use both together to maintain the high photostability. Then the question is, now that we have argued that these single and double-strand polymers are highly photostable, so they have different mechanisms as shown here from high energy states to maintain the photostability, can nature, or was nature able to use this high energy state to perform some work? In particular, can nature have used this different photostability mechanism to potentially repair themselves? And so to introduce that concept, I first have to show that when you have two stacked bases, a particular timing bases as shown here, 1 in every 100 photons, or number of high energy ultraviolet photons, can induce a chemical reaction. So this reaction is extremely small. It's only 1 in 100 photon that happens, or less than 1%. But when the bases are in the right conformation, in the stack conformation as shown here, a reaction which is called a dimerization can happen, which if it's not, it damages the DNA or the RNA strand. And so in the absence of enzymatic repair mechanism that was not available during the prebiotic era, then it begs the question how the RNA and the polymers may have survived these chemical events. And so surprisingly and really remarkable, science has shown that in these polymers, which you can consider as primordial polymers, not the final DNA and RNA polymers that we have nowadays, but polymers that also include modified bases, or bases that have a different composition, all these modified bases can absorb ultraviolet radiation, and then they can repair the damage of the strand. And so it is now believed that in the prebiotic era, primordial polymers, RNA and DNA polymers that include other modified bases, could have used the modified base as a self-repair mechanism to repair any damage that it was caused by ultraviolet radiation without the need of using enzymatic repair mechanism. Okay? And so this figure summarizes the presentation, and so to conclude, I want to highlight that I have emphasized on the role of ultraviolet radiation from the sun that may have played in shaping the chemical inventories during the prebiotic Earth. And this information also is potentially important to understand to obtain insights about what kind of molecule have potentially been used in other planets as the building block of life. I also have argued that neutralizing these high energy states in the monomers, in the base pair, in the base stack, and in the double helix is a very effectively pathway to maintain photostability, but when they also can be used by nature as a self-repair process to maintain the stability of any chemical damage in the DNA in the absence of enzymatic repair mechanism. And so now I will be happy to answer any questions that you may have. Thank you.
The Origin Science Scholars Lectures are presented by Case Western Reserve University's Institute for the Science of Origins, with the assistance of the Siegel Lifelong Learning Program, the College of Arts and Sciences, and MediaVision. For more information on the Origin Science Scholars Program, including a full video archive, please visit the Institute's website at origins.case.edu.